Okay, so welcome back to this next video in which we are discussing the intestinal stem cells. Okay, right, so we are in the process of discussing the Wnt beta-catenin pathway, because remember, it is this pathway which is going to maintain a intestinal stem cell as an intestinal stem cell. Okay, so we want to see how the Wnt secreted by the Paneth cells at the base of the crypts of the epithelium maintain intestinal stem cells as intestinal stem cells. So we've seen that what's going to happen is that the Wnt molecules are going to bind to the amino terminal domains of frizzled receptors, and they're also going to bind to binding sites on a co-receptor, which is either LRP5 or LRP6, okay? And this complex now is going to activate the Wnt beta-catenin pathway. Now, basically, the Wnt beta-catenin pathway is often modeled as having two states. There is the off state of the Wnt beta-catenin pathway, which is the state where there is no Wnt present in the extracellular fluid. Okay, so there is no activation of the frizzled receptors and the LRP5/6 receptors. And then there is also the on state, which is the state when there is Wnt in the extracellular fluid, and therefore there is the activation of the frizzled receptors and the LRP5/6 receptors. Okay, so we are going to start off by discussing the off state, and then we will discuss the on state. So at the moment we're going to take a step back and discuss what is happening in the cell when there is no extracellular Wnt, and therefore there is no activation of the frizzled receptor or the LRP5/6 co-receptors. Okay, right, so let's get a new piece of paper and discuss what is happening in the Wnt beta-catenin pathway when it's in the off state like so. Okay, right, so in the off state you have assembled a complex of proteins which is going to destroy beta-catenin. Okay, so I think we firstly probably need to introduce beta-catenin itself. So beta-catenin is a protein, and it's going to act as what's known as a transcriptional activator, but we'll come to what that actually means much later. Okay, so here is beta-catenin. Here is its amino terminus, and here is its carboxylic acid terminus. And we'll colour in beta-catenin in red. Okay, right. So we're now going to discuss the fact that usually in the off state of the Wnt beta-catenin pathway, beta-catenin gets made, and then the instant it gets made, it's going to be destroyed. And the reason for this is that during the off state, you have assembled structures known as beta-catenin destruction complexes. Okay? And these are complexes of multiple proteins which are going to be involved in the destruction of the beta-catenin. Okay, so we now want to see which proteins are going to be involved in this beta-catenin destruction complex and what it is actually going to do to the beta-catenin that is going to facilitate the destruction of the beta-catenin. Okay, right. So basically, the first component of this beta-catenin destruction complex that I want to discuss is a scaffold protein known as axin. Okay, so this rectangle here, this is going to represent the scaffold protein axin. Okay, so I'm going to colour this protein in orange. So a scaffold protein is just a protein whose function is just to bind to other proteins. Okay, so axin here is an example of a scaffold protein. Okay, right. Now, the axin is actually going to bind to another scaffold protein to start the formation of this beta-catenin destruction complex. So another scaffold protein is going to bind here, and this second scaffold protein is what's known as APC. Now, APC stands for Adenomatosis polyposis coli. So let me just move this over a little bit. So the A is for adenomatous, okay, the P is for polyposis, okay, and then the C is for coli. Now, this is named after a hereditary condition which predisposes you to developing colorectal carcinoma, which involves a mutation in this protein. Okay, right. So APC, or adenomatous polyposis coli protein, is another scaffold protein. So it's just a protein which binds to other proteins, and its role is in holding other proteins together, two or more proteins that are different together, basically, in a complex. So the start of the beta-catenin destruction complex consists of these two scaffold proteins bound together. Okay, now we're going to actually get some enzymes binding onto these scaffold proteins. So the first one I'm going to mention here is called glycogen synthase kinase 3 beta, or GSK3 beta for short there. Okay, so I'll write this out here. So that enzyme there is GSK3 beta, and this stands for glycogen, that's the G, okay. Then the S is for synthase. So this is glycogen synthase, and then it's glycogen synthase kinase, that's the K. So this is glycogen synthase kinase, specifically glycogen synthase kinase 3 beta. Okay, so we'll colour glycogen synthase kinase 3 beta in green here. Okay, so this is glycogen synthase kinase 3 beta. I think I'll also underline adenomatous polyposis coli in vivid purple. Okay, right. Now, this is a kinase enzyme, okay? It's specifically a serine threonine kinase. So I just want to discuss what a serine threonine kinase actually is, because we haven't discussed this at any point previously in this video. So I just want to make sure everyone is on the same page with an understanding of what a serine threonine kinase is going to do. Okay, right. So a serine threonine kinase enzyme is an enzyme that is capable of phosphorylating serine and threonine residues within proteins. So let me discuss the structure of a serine and a threonine residue. So let's draw out the structure of a serine residue and a threonine residue. So we're going to draw them as residues, not as free amino acids. Now what that means is that we are going to draw them as they would appear within a polypeptide, rather than drawing the free amino acid. So here is the amino group of this amino acid residue, as though it's bound to the carboxylic acid group of the amino acid prior to it. Then you've got an alpha carbon with a hydrogen coming off it, and then the carboxylic acid group here, which will be bound to the amino group after it. So we're not drawing the free amino acid, we're drawing the residue, which means we're showing it as though it's within a polypeptide, like so. So this is the core amino acid residue structure. Okay, this is common to all amino acid residues. Now the R group of a serine residue is going to consist of a methylene group, like so, with an alcohol group coming off it. Okay, so this is the structure then of a serine residue. Okay, let's see how threonine compares to this now. So here is the amino group once again. So we'll start by drawing the core amino acid structure firstly. Here's the amino group, here's the alpha carbon with the hydrogen coming off it. Here's the carboxylic acid group. And once again, we're drawing a residue, so I've drawn it as though it's bound to two amino acids, one prior to it and one after it. Now the R group of a threonine residue consists of a carbon, once again with an alcohol group coming off it, and also a hydrogen, but this time it's also got a methyl group coming off it. Okay, so this is threonine, and we can see that threonine is almost identical to serine. Okay, so these are the residues that we are going to phosphorylate then. So what does a serine threonine kinase now do? It's going to stick phosphate groups onto these serine and threonine residues. Now, serine threonine kinase enzymes are going to take phosphate groups from ATP molecules and stick them onto these serine and threonine residues, reducing the ATP molecule to now being an ADP molecule. Okay, now that's quite complicated to show, so instead what I'm going to show is I'm going to show how we could bind an inorganic phosphate molecule to the alcohol group of a serine and threonine residue. This is not actually what the serine threonine kinases are going to catalyze. They're not capable of catalyzing that, okay? But it will get the principle of what it means to phosphorylate a serine and threonine residue across. Okay, so the structure of an inorganic phosphate molecule then consists of a phosphorus atom, double bound to an oxygen atom here, with 2 alcohol groups coming off it, and then a single bond to an oxygen atom, which has then acquired an electron via ionic means and therefore has a negative charge on it. Okay, now you can bind inorganic phosphate molecules like this to alcohol groups on molecules such as the serine or threonine residues. Now, the reason is that if you look at this group that I have circled in red here, where we've got the phosphorus atom double bound to an oxygen atom with an alcohol group coming off it, that looks extremely similar to a carboxylic acid group. In fact, if that phosphorus atom was a carbon atom, that would be a carboxylic acid group. Okay, so what we can actually do is we can bind this carboxylic acid-like group to the alcohol group in pretty much the same way as a carboxylic acid group would bind to an alcohol group. Okay, so when a carboxylic acid group reacts with an alcohol group, it forms an ester link. We're going to form something very analogous. We're going to take the alcohol group of the phosphorus atom, and we're going to take the hydrogen off the alcohol group. We're going to combine those two things together to make water. We're then going to bind the oxygen atom of the alcohol group of the serine or threonine residue to the phosphorus atom of the inorganic phosphate molecule. And this link that we've created here, this, to show its parallels with an ester link, we call this a phosphoester link. Okay, so this is what you overall form when you phosphorylate serine and threonine residues. You link this phosphate group here onto the alcohol groups of serine and threonine residues via phosphoester links. Okay, so that the product of this reaction is the same as the product of the reaction that these serine threonine kinases are going to catalyze. It's just we don't get the phosphate group from an inorganic phosphate in the case of these serine threonine kinases. We get it from an ATP molecule. Okay, but it gets the principle across of what you're going to do. You're going to phosphorylate the alcohol groups of serine and threonine residues. Okay, right, and this can change the properties of proteins hugely. Okay, so that's what glycogen synthase kinase 3 beta is then. It's a serine threonine kinase enzyme, and we're about to see another serine threonine kinase. So, glycogen synthase kinase 3 beta is attached both to axin and also to Adenomatosis polyposis coli, but this next serine threonine kinase is only going to be attached to axin. Okay, so here it is in blue here. And this is casein kinase 1 alpha, okay, which I'll abbreviate to CK1 alpha. So CK1 alpha stands for casein, that's the C, and then kinase, that's the K, and then it's casein kinase 1 alpha. Okay, so this is the enzyme we've got there in blue. Right, so this complex of these 4 separate proteins, axin, Adenomatosis polyposis coli, glycogen synthase kinase 3 beta, and casein kinase 1 alpha, this now is the full beta-catenin destruction complex. Okay, so in the off state of the Wnt beta-catenin pathway, when there is no Wnt in the extracellular fluid, you have these monsters floating around in the cytoplasm, and the instant beta-catenin molecules get made, okay, so the instant they finish and they're out of the ribosome, they are going to bind to these beta-catenin destruction complexes. So let's show that here. Okay, so the beta-catenin molecules bind to both the axin scaffold protein here and also to the Adenomatosis polyposis coli scaffold protein. So it binds to both of them here. The amino terminal end binds to the axin scaffold protein, and the carboxylic acid terminal end binds to the Adenomatosis polyposis coli scaffold protein. So this is beta-catenin in red here. Okay, right. So, when the beta-catenin binds to the beta-catenin destruction complex, it's now going to be phosphorylated on serine and threonine residues by our two serine threonine kinases. Oh, and I should have mentioned, Casein kinase 1-alpha is also a serine threonine kinase, just as glycogen synthase kinase 3-beta is. Okay, right, so what's now going to happen is Casein kinase 1-alpha is going to begin the phosphorylation of the beta-catenin. Okay, so the first phosphorylation is going to be undertaken by Casein kinase 1-alpha. Okay, so it is going to phosphorylate a serine residue within the beta-catenin molecule. Specifically, it's going to phosphorylate serine 45. Okay, so that means if you start at the first amino acid and count along the polypeptide, count amino acids, when you get to the 45th one, that will be a serine, and that residue is going to be phosphorylated by Casein kinase 1-alpha. Okay, and that's the first thing that happens. Okay, this is known as the priming phosphorylation. Okay, so we say that Casein kinase 1-alpha is going to prime the beta-catenin so that it can now be phosphorylated further by the glycogen synthase kinase 3-beta. So that's all Casein kinase 1-alpha is going to do. It's not going to phosphorylate any further. The rest of the phosphorylations that are going to occur are done by glycogen synthase kinase 3-beta. Okay, so there are 3 further phosphate groups that are going to be stuck onto serine and threonine residues. Okay, so I'll colour these in here. So these 3 are 3 more phosphate groups. Now, they are going to be added on to threonine 41, okay, which is that second one here. Okay, then we've got serine 37. Okay, that's the third one. So we're getting gradually closer to the amino terminus. And then finally, also serine 33. So those are the 3 residues that glycogen synthase kinase 3-beta is going to phosphorylate on the beta-catenin molecule, but only once Casein kinase 1-alpha has phosphorylated serine 45. So that first phosphate group added on by Casein kinase 1-alpha primes the beta-catenin so that glycogen synthase kinase 3-beta can now phosphorylate it at these 3 further sites. Okay, right. Then what's going to happen is the beta-catenin that has been phosphorylated 4 times is now going to leave the beta-catenin destruction complex. And now, once it's been phosphorylated like this, another enzyme can bind to it. Okay, so let's draw our beta-catenin here. So here's the amino terminus, here's the carboxylic acid terminus, and now we've got these 4 phosphate groups added onto the beta-catenin molecule. Okay, so we'll colour these in purple here. Right, okay, so now what's going to happen is an enzyme is going to come and bind here. But I think firstly I'll just colour in the beta-catenin in red here. Okay, so what's going to happen is an enzyme is going to come and bind to these this phosphorylated beta-catenin, and it can only bind once the beta-catenin has been phosphorylated like so. Okay, and this enzyme is what's known as beta-TRCP. Okay, so we'll colour in beta-TRCP in green here. And this is an enzyme which is capable of adding ubiquitin groups onto proteins. Okay, so now what's going to happen is the beta-TRCP is going to stick a ubiquitin group onto the beta-catenin. Okay, so I'll show this by this square group here. Okay, and we'll colour in this ubiquitin group in blue. So once you've got beta-TRCP bound to you, it is going to stick this ubiquitin group onto you. Now, proteins do not want ubiquitin groups stuck onto them, because once a protein has got ubiquitin stuck onto it, it's not good news for that protein. It's going to get targeted to a truly terrifying structure in the world of molecular biology, okay, called the proteasome. So let me draw this here. So within the cytoplasm of cells, you have a structure called the proteasome, okay, and basically proteins go in one end and amino acid fragments, polypeptide fragments come out the other end. So the proteasome is a tube, okay, and you're going to feed your polypeptide in this end, and as it goes through, it's going to be broken down by a huge number of different enzymes. They don't quite break it down into amino acids. They break it down into fragments of a few amino acids then. Okay, now how does a protein end up going in here? Well, basically, ubiquitin binds to the entrance of the proteasome. Okay, so here's the ubiquitin group. It will bind to the entrance of the proteasome, and then it will guide the polypeptide into the proteasome, and the polypeptide will get poured in, basically. Okay, so here is our beta-catenin going into the proteasome, basically. Okay, and it will still have those phosphate groups on it as well, so let's put those on. Okay, right, so that then is how we're going to destroy beta-catenin. So normally, in the off state of the Wnt beta-catenin pathway, the beta-catenin destruction complexes are perfectly functional, okay? They are going to phosphorylate beta-catenin as soon as it's produced. That's going to allow the beta-catenin to bind to beta-TRCP, which will then ubiquitinate it. The ubiquitin will then target it for destruction by the proteasome. So basically, in the off state of the Wnt beta-catenin pathway, you have no beta-catenin floating around within the cytoplasm. Okay, right. In the next video, what we'll see is once you go into the on state, i.e. once Wnt is present in the extracellular fluid, and you have got the activation of the frizzled receptor along with the LRP5 or the LRP6 co-receptor, then we're going to get the inactivation of beta-catenin destruction complexes, and beta-catenin is going to go up within the cytoplasm of the cell.